This is a revision video on kidney structure and function. So this video covers all aspects of the kidney required knowledge. So it goes into detail in terms of understanding the nephron and each component of the nephron and how it works. I also recommend that you review knowledge from previous topics that are relevant here. That include osmosis, tissue fluid formation, and the countercurrent flow system in fish gills. All those pieces of knowledge are important for understanding here. As always, there are revision questions, so in this case the 13. So attempt the questions and then you can review your answers. Because this video is quite detailed, in the descriptions, timings for the relevant parts are provided. So if you wanted to skip to a specific part, such as the structure of the nephron, you can skip straight to that. Otherwise, you can continue with the whole of the video. Okay, so pause the video, attempt the questions, and then answers will follow. Okay, so to begin with, we deal with the kidney itself, or the gross structure of the kidney. So question 1 was asking about function. So broadly, there are two functions. So in terms of excretion, that's removal of nitrogenous waste, mainly in the form of urea. And then osmoregulation is the regulation of the osmotic potential of blood and tissue fluid. So, as in the questions, you could have drawn your own diagram and labelled it, but I've provided a diagram here. So if you wanted to use this diagram, then there are numbered labels to fill in, but you should have the following structures. So they are the key components. At this point, we can focus on an area of the kidney, for instance, this area, and that includes part of the cortex and the medulla. And if we magnified that area, we would find a specific structure that we're going to look at in more detail next. That is the nephron. And just for you to be aware that there are approximately 1 million nephrons per kidney. So let's move on to questions 3 and 4, and we'll look in more detail at the nephron. So, as before, you could have drawn this yourself and labelled it. I've also provided a diagram here with numbered parts. So let's go through the key components you should have on your diagram or have labelled from this diagram. So firstly, we know one part of the nephron is found in the cortex, and then another part is found extending into the medulla. And so this is an example of a nephron with the loop of Henle that's extending into the medulla. In humans, about 20% of the nephrons look like this, are nephrons that extend into the medulla. The remaining 80% have much shorter loop of Henle, and they really only occupy the cortex. And the nephron approximately is about 3 centimeters long. And as we mentioned in the previous diagram, there are about 1 million nephrons per kidney. Okay, so let's move on to the first detail that we're going to look at, which is ultrafiltration. And that obviously occurs in the Bowman's capsule and glomerulus. So you could have drawn and labelled this yourself. Alternatively, there is a diagram here for you to use to add labels to, so I will provide those answers here. So just to note that to help you out with remembering the afferent arteriole and the efferent arteriole, I think the easiest thing is to remember that A comes before E in the alphabet, and so that should help you remember that the afferent arteriole enters the Bowman's capsule, and the efferent arteriole is the one that leaves. Okay, so there are a few structures that we're missing here, so let's add those in. And so hopefully what you're able to identify is that 7 and 8 are and that podocyte has these extensions that extend around the endothelial cells. And obviously they're involved in this process of ultrafiltration, really important cells. So how is the efferent arteriole and afferent arteriole different from each other? So you should be able to state this. So importantly, the lumen of the efferent arteriole is narrower than the afferent arteriole. And so the consequence of that is that there is increased hydrostatic pressure in the glomerulus, and this leads to this process. And I've underlined spaces between, so it's not that the water, urea, glucose, and mineral ions go through those cells, they actually go through spaces between them. And what's formed is glomerular filtrate. So what cannot get through during ultrafiltration? So red blood cells and plasma proteins that are too large to leave. Okay, so let's move on to the next component of the nephron. So filtrate from Bowman's capsule moves through the proximal convoluted tubule, and this has a series of capillaries in close association with it. So if we focus on a small component of the wall of the proximal convoluted tubule and that blood vessel that flows next to it, we would see something like this. And so those three events are depicted here in the diagram. So you should have stated that the three events that lead to absorption of substances back into the blood are these three events. And this should be familiar from the digestion topic and the way that glucose and amino acids are absorbed from the ileum into the blood. Notice that these cells have microvilli. Obviously, they increase the surface area over which absorption can occur. And so the four substances that are absorbed into the blood from the proximal convoluted tubule, for examples, are glucose, amino acids, chloride ions, and the fourth, and importantly, is water. So water by osmosis passes from the filtrate into the blood, and about 85% of the water in the filtrate can re-enter the blood from the proximal convoluted tubule. So from here, filtrate moves to the descending loop of Henle. So let's take a more detailed look at that. Before we take a closer look at the loop of Henle, I want to just have a brief discussion about osmotic concentration. So you'll see the osmotic concentration used specifically in the loop of Henle, and I just want to ensure that you understand that. So let's deal with firstly just the basic idea of concentration. So commonly concentration of a substance is expressed as moles per litre. And that can actually be abbreviated to molar. So 1 mole per litre is equal to 1 molar. And so let's use an example of a substance. So we've got a water molecule here, our solvent, and let's say we have a solute, glucose. In our example, glucose could be at 0.005 molar. We could express that as 5 millimolar, milli being 10 to the minus 3. Let's also assume that we have some other solutes in there. So here we've got some sodium ions. In our example, there are 140 millimolar. And let's say we've got some urea, 3 millimolar. So molecules all relevant to the filtrate and formation of urine. So in this example, with our solutes, we've considered them individually and their individual concentrations. But when we're considering the osmotic effect of these molecules in a solution, we consider them all together, and we express that as osmoles. So osmoles per litre, or we could also say osmolar. So in our example, if we consider them all together, we can consider the osmotic effect of all of those solutes. We might express that as 300 milliosmolar. That is the osmotic impact of our solution here. This is a convenient example because plasma actually has a osmotic concentration of about 300 milliosmolar. Human urine, usually around 900 milliosmolar, but just as a caution, that can be between 50 and 1,400 milliosmolar, depending on fluid intake or fluid loss. And just to note then that human urine has a lower water potential than plasma. So the higher the osmolar value, that indicates a lower water potential. So that's just really important to pay attention to. Okay, so now we've established that, let's move on to looking at the loop of Henle. Okay, so filtrate enters from the proximal convoluted tubule. The fluid in between the descending limb and the ascending limb of the loop of Henle is interstitial fluid. And then around and surrounding the loop of Henle are capillaries carrying blood. So how does the permeability of the walls of the loop of Henle change along its length? Well, if we look at the descending limb, and just to note, it's slightly permeable to sodium ions, whereas the ascending limb, and this is really important for how it functions. So let's now look in more detail at the movement of sodium ions and water in and out of the loop of Henle descending limb and ascending limb. So we'll do it in a step-by-step process, and you can check your answer against that. So filtrate entering will have a value of approximately 300 milliosmolar. And what's really important here is that the ascending limb actively transports sodium ions out into the interstitial fluid. As a consequence, that increases the sodium ion concentration. There's some small amount of sodium ion diffusion into the descending limb, but as a consequence of this, the water potential in the interstitial fluid at this top region, the region nearer the cortex, is lowered. And so water diffuses out of the descending limb by osmosis, and then from the interstitial fluid it moves into the blood by osmosis and is carried away. That obviously lowers the concentration, or the osmolar concentration of the filtrate, and as a consequence lowers the water potential. And as we move down, sodium ions are actively transported out, so that increases the sodium ion concentration in the interstitial fluid. Some sodium ions move in to the descending limb, lowering the water potential of the interstitial fluid even more. And then water diffuses out of the descending limb by osmosis and carried away in the blood. And this carries on right down the ascending limb and descending limb, so this process continues. And so as you can see, that green colouring, which depicts sodium ion concentration, increases the further down the loop of Henle you get, and the more into the medulla you get. And so the filtrate that's right at the bottom of the loop of Henle is at quite a high milliosmolar concentration because it's lost the majority of its water into the interstitial fluid, which has been carried away in the blood. What you'll also notice is that right at the bottom of the loop of Henle, sodium ions can diffuse back into the interstitial fluid just because of those concentration differences that have been created. And so that further lowers the water potential of the interstitial fluid. And then, so as that filtrate then moves back up the ascending limb, because sodium ions are gradually moved out by active transport of that ascending limb, but water can't move out, it's impermeable to water, remember, the osmolar concentration is reduced in the filtrate, but the water potential increases. So by the time the filtrate reaches the top of the ascending limb, it has a much lower milliosmolar concentration, around 200, and a much greater water potential. And from there it moves into the distal convoluted tubule. So let's take a closer look at the distal convoluted tubule. So filtrate from the loop of Henle enters the distal convoluted tubule, and this looks very similar to the proximal convoluted tubule. It's got blood vessels associated with it, but let's take a closer look at the wall of the distal convoluted tubule and how it functions. So the cells of the wall of the distal convoluted tubule have microvilli to increase the surface area for absorption, has many mitochondria, and that's for good reason. So the three functions of the distal convoluted tubule should have stated are, so obviously that's why it requires many mitochondria. And so that's about balance of hydrogen ions and bicarbonate into the blood. And so water reabsorption is obviously a key function of the kidney that occurs here also, and that's dependent on aquaporins. So these are protein channels for water that are found in the cell membrane of the cells that line the distal convoluted tubule. And so the more of those there are, the more water can diffuse by osmosis back into the blood. Okay, so finally we'll move on to the collecting duct. So let's just take a quick look at the collecting duct before we answer the questions. So filtrate enters the collecting duct from the distal convoluted tubule. Surrounding the collecting duct is the interstitial fluid. So the filtrate that enters is at around 300 milliosmolar. That's similar to blood plasma. And remember that the loop of Henle is in close proximity to the collecting duct in the medulla. And remember from what we discussed about the way the loop of Henle functions, there is a sodium ion concentration gradient that increases from the cortex down into the inner medulla. And so if we consider the water potential of the interstitial fluid going down from the upper medulla, the cortex end of the medulla, down into the inner medulla, it looks something like this. So there's a gradually reducing water potential. And so this is where the counter-current multiplier mechanism is really important. So we'll take a look at it in diagrammatic form, and then I will give you a text answer. So at the top of the collecting duct, the water potential in the interstitial fluid is more negative than the filtrate inside the collecting duct. So water diffuses out by osmosis and then is carried away in blood vessels in the area. That reduces the water potential in the filtrate and increases the molar osmolarity. That meets or runs next to interstitial fluid where the water potential is more negative. So again, water moves out by osmosis, carried away in the blood. And so that process continues the whole way down the collecting duct. And that results finally in this production of concentrated urine that contains waste products, nitrogen-based waste products such as urea and any drug metabolites that haven't been reabsorbed. And obviously they move into the renal pelvis, move out of the kidney via the urethra and reach the bladder. And so in text form, that countercurrent multiplier mechanism is described here. And what you should remember from that countercurrent information from the way the fish gill functions, and it's the same here, is that there's a water potential gradient between the collecting duct and the interstitial fluid along the whole length of the collecting duct. There is not a large gradient, water potential gradient, between the collecting duct and the interstitial fluid. It is always a small water potential gradient, but it is enough to ensure that there is a continual movement of water out of the collecting duct along the whole length. That's really important. That's what makes this process efficient. And so finally, what determines the permeability of the collecting duct wall to water? That's the concentration of the aquaporin channels also found in the distal convoluted tubule. And that's regulated by antidiuretic hormone. Okay, so that is kidney structure and function. It's a lot of information, so I strongly encourage you to review it regularly. Try these questions. Any of the components that you are struggling with, then you can skip directly to those using the time signatures in the descriptions below.